Applied Biology, and this is a lecture on enzymes. And enzymes are proteins. That's the first thing that you need to put in your PowerPoint. Proteins, remember, are one of the four kinds of organic macromolecules that we have just studied. And enzymes are a specific example of proteins, and they're a particularly important example because they are the workhorses, the molecules that get jobs done within a cell, whether it be building things, building molecules, or breaking molecules. Okay? So enzymes are proteins, and enzymes speed up reactions without being destroyed in the process of those reactions. That might sound familiar. That's because enzymes are biological catalysts. Catalysts are molecules that, yes, they increase the speed of reactions without being used up in the reaction, and you learned about those in chemistry last year. Enzymes are a specific kind of catalyst that operate within biological systems. Here's your review of the definition of a catalyst: a chemical agent that changes the rate of a reaction without being consumed during the reactions. So each enzyme catalyzes a very specific reaction, and because there are thousands of reactions that need to happen within cells, there are thousands of enzymes that need to be present to make those reactions go at a speed that will keep the cell alive. Okay? The last thing is that enzymes have suffixes with the letters ASE, pronounced ACE. Okay? So for example, the enzyme that we are going to be using in the lab this week is called hydrogen peroxidase, ending in ASE. Okay, so this graph should look familiar to you, again, from your study of catalysts in chemistry, but I just want to review the reaction pathway graph. Okay? First of all, I want to review what the energy of activation is. Well, actually, before I do that, let me just review what belongs on the x and the y-axis. So, on the y-axis, we have energy. Okay, so it's not labeled here, so you may want to label on your PowerPoint. Energy is the y-axis label, and the unit for energy is joules. And label your x-axis. It says reaction coordinate down here, but let's label it time. And this would take place in seconds, or it may even take place in milliseconds. Seconds may be too long. Okay? So here is our reaction. It's going to take place over time. Our reactants start out at a certain level of energy. They need to be bumped up to the energy of activation before the reaction can start. Your energy of activation is defined over there on the right. That is the energy needed to break the bonds in the reactant molecules so that they can recombine and form the products. So this is the energy to start the reaction. Okay? The Gibbs free energy, or delta G, is the change in energy between reactants and products. Okay, we're going to look at a couple different examples of Gibbs free energy. In this case, the reactants have a higher energy than the products, so we have a negative delta G between the reactants and the products, a loss of energy from the molecules out into the system, and this negative delta G would cause this reaction to release heat. Okay. So let's take a look at another similar reaction, but this time let's add an enzyme to it. Okay? Again, we have our free energy in joules out here on the y-axis, the course of the reaction or time on the x-axis. This is our normal reaction pathway, and this is our enzyme catalyzed pathway. If you think of these graphs like a hill, and you were asked to push a ball over each one of these hills, I assume that you would choose this red hill. It's a lower hill. It happens much more easily than the blue hill. So each time there's a chance of this reaction happening, it happens much more quickly when it is catalyzed. But notice that the enzyme is not used up in the course of this reaction. The reaction just happens more easily. Okay? Also notice that the enzyme does nothing to change the energy of the reactants, starts out at the same place, same energy level, and it does nothing to change the energy of the products. So it ends up at that energy level. I want to look at one more graph with you, okay? Because the last two graphs that we looked at were exothermic reactions, that is, they released energy into the environment from the reaction, and I want to look up here at an endothermic reaction. In this case, again, free energy and time on the axes, but in this case, the initial state or the energy of the reactants is lower than the final state or the energy of the products. So in this case, you have a very large energy of activation, whether it's uncatalyzed or catalyzed, but the catalyzed is smaller, and you end up with more energy in the products than you had in the reactants, and therefore energy is absorbed from the environment, heat is taken in, and if you were holding a beaker with an endothermic reaction happening, it would feel cold. Either way, whether it's endothermic or exothermic, the job of the enzyme is to decrease the energy of activation.